Welcome back, my dear students. Today we will explore an important cell organelle, the Golgi complex, often called the cell's packaging and dispatching center. It plays a vital role in modifying, sorting and transporting proteins and lipids within the cell. Let's understand its structure, major functions and overall significance in maintaining cellular organization and activity in a simple and clear way. Golgi apparatus as a component of the GERL system. GERL system basically stands for Golgi endoplasmic reticulum lysosome system. It represents a functional unit involved in the synthesis and modification, packaging and transport of cellular materials. The Golgi apparatus plays a central role in this system by acting as an intermediate station between the endoplasmic reticulum, ER and lysosome. In our previous class we have learned about the endoplasmic reticulum, and in our next class we must learn about lysosome. So Golgi apparatus acts as an intermediate between endoplasmic reticulum and lysosome as a component of the GRL system. Golgi apparatus was discovered by Camillo Golgi in 1898 from the nerve cells of an owl. It was first seen by L. V. St. George in 1867, and firstly it was observed by Dalton and Felix in 1954 under electron microscope. Golgi apparatus is also known as Golgi complex or Golgi bodies, Golgi zome or Golgi membrane. In plants they are known as dictyosome. Golgi apparatus also called as Golgi stack, lipochondria, Dalton complex, Becker's bodies, etc., and they can be treated as carbohydrate factory or traffic police of the cell. Organization within cells. In animal cells it is usually a single compact structure, and it has two conditions: localized type and diffuse type. The localized type, they are common in most vertebrate cells, and diffuse type, it is found in invertebrate and invertebrate liver and nerve cells. Position between the nucleus and cell membrane. In plant cells it consists of many unconnected units called dictyosomes or Golgi stacks. Each dictyosome has its own surrounding zone of exclusion or a clear cytoplasmic area. Polarity of Golgi apparatus. Basically Golgi apparatus has two distinct phases: cis phase or forming phase, which is a convex surface. It faces the nucleus or endoplasmic reticulum or ear. It receives transport vesicles from ear. It also called as cis Golgi network or CGN, and membrane thickness of the forming phase or cis phase is nearly 4 nanometer thinner. Trans phase or maturing phase, concave surface. It faces the cell membrane, buds of secretory vesicles and vacuoles. They are also called as trans Golgi network. Membrane thickness of the maturing phase or trans phase is about 7 to 8 nanometer thicker. Occurrence: Golgi complex or Golgi apparatus is present in all eukaryotic cells, both plants and animal cells, but they are absent in gametes of bryophytes and pteridophytes, male gametes of bryophytes and pteridophytes, mature sheep tubes, some fungal cells, mature sperms and RBCs of animals, and in all prokaryotic cells. Their number varies, usually 1 in simple alga like Micromonas and up to 25,000 in rhizoid cells of Cara. Liver cell up to 50 dictyosomes. They occupy about 2% of total cell volume. Shape and size. Shape and size vary depending on the physiological state of the cell. Average size in plants in between 0.5 to 1 micrometer in diameter. The structural components include a typical Golgi apparatus is usually made up of 4 main parts like cisterna, tubules, vesicles and vacuoles. Cisterna, it is basically consists of 4 to 8, it can be 3 to 20 flattened smooth membrane bound sacs. Each saccule encloses a lumen of 60 to 90 angstrom. Adjacent cisterna are separated by 100 to 300 angstrom. The lumen contains a fluid or matrix that processes biomolecules. Cisterna show definite polarity, that is convex cis and concave transite. The substances move from cis to trans phase through subtle vesicles. These are the microvesicles. There are vesicles. Tubules. Tubules form a network mainly near the periphery and trans phase. They arise from the fenestrations or opening in cisterna. Their diameter is about 30 to 50 nanometer. Their function, they interconnect different cisterna and assist in transportation. Vesicles, they're small spherical sacs, diameter about 20 to 80 nanometer. They're found attached to the tips of tubules. There are two types of vesicles: smooth vesicles contain secretory materials or secretory vesicles, and coated vesicles. They are covered by clathrin, triskelion protein involved in membrane protein transport. Golgi vacuoles, they are enlarged or swollen parts of cisterna formed at the transphase. Contain granular or amorphous material, and some act as lysosomes. Vesicular transport. Transport or subtle vesicles, they move materials between cisterna that is seized to transphase. Secretory vesicles border from TGN or trans-Golgi network carrying glycoproteins, glycolipids and polysaccharides to various destinations or outside the cell. Large secretory vesicles are called as Golgian vacuoles. Special features: they stain selectively with neutral red. They does not take Jenna's green, which stains mitochondria. During cell fractionation or centrifugation, mitochondria settle fast as it is heavier, but Golgi bodies settle later as it is lighter. So presence confirmed in both plants and animals using electron microscopy. It was discovered by Camillo Golgi in 1898. Main function is the collection, modification, packaging and secretion of biomolecules. Their polarity: cis or forming phase and trans or maturing phase. Components include cisternae, tubules, vesicles and vacuoles. Staining: neutral red positive but Jenna's green negative. It acts as traffic police or carbohydrate factory of the cell. Functions of Golgi apparatus. The Golgi apparatus is basically a cellular organelle that processes, modifies and packages proteins and lipids synthesized in endoplasmic reticulum. It plays a pivotal role in post-translational modification, adding carbohydrates and other molecules to proteins to create functional structures. The Golgi apparatus sorts and packages these molecules into vesicles, which then transport them to specific cellular destinations or release them outside the cell. Additionally, it contributes to the formation of lysosomes, organelle containing enzyme for cellular digestion. Through these functions, the Golgi apparatus ensure proper protein and lipid functionality, distribution and cellular maintenance. So my dear students, today we have learned how the Golgi complex acts as the cell's packaging, modifying and dispatching center, ensuring that proteins and lipids reach their correct destinations. From forming lysosomes and acrosomes to help in secretion and membrane formation, the Golgi plays a key role in maintaining cellular organization and efficiency. If you enjoyed understanding this vital cell organelle, don't forget to like, share and subscribe for more fascinating lessons in cell biology. See you in the next class. Thank you.